Now that we've taken a look at the structure of nitrogenase, including the two components, the iron protein and the MOFI protein, now we have a structural basis with which we can discuss the mechanism. And so, to be clear, there's been quite a bit of work, mechanistic work done on nitrogenase. So we'll only be able to cover some of the aspects due to time constraints. But here are some of the mechanistic questions, although this is not a comprehensive list. For instance, how are electrons delivered from the iron protein ultimately to FeMoCo, which is the active site for the nitrogen reduction? What is the importance of protein conformational changes? What N2-based intermediates are made? How is the energy from ATP hydrolysis transduced? And when and how is hydrogen made? So like I said, this is not a comprehensive list of mechanistic questions that have been studied or yet to be resolved for nitrogenase, but these are some of the ones that we're thinking about right now. In particular, what we'll talk about right now are the ones highlighted in green in the next few slides here. Okay. So first, let's take a look at the electron transfer. So let's go back to this schematic where the blue ovals are showing the iron protein, and then the red and orange rectangles are showing the two subunits for the MOFI protein. Okay, and then within the iron protein, there's a green cube. Okay, this is indicating that if you look at the legend, this is indicating that the iron sulfur cluster within the iron protein is reduced. Okay, so it's in the +1 state. In the MOFI protein, the two cofactors, the P cluster is shown as a rectangle, and in the gray color, this is a neutral. This is in the neutral oxidation state. The diamond is the active site, or the FeMoCo, and again, as gray, this is in the neutral oxidation state. So electrons need to be passed, remember, from the iron sulfur cluster of iron protein to the P cluster, and then ultimately to the FeMoCo, which is the active site. So we're going to look at several steps of this pathway, and I just want to note now that if you see a change in shape in the depictions, so for instance, at some point the rectangles change to more rounded rectangles, that indicates a change in the protein conformation, or in other words, the protein shape. Okay, so the first thing within this mechanism is that the reduced and ATP bound iron protein is now primed to dock with the MOFI protein. So only in the state where where ATP is bound to both of the sites on the iron protein and the iron sulfur cluster is reduced, is it allowed to associate with the MOFI protein. So docking occurs here, and then what happens is is somewhat counterintuitive. So the first electron transfer actually doesn't occur from the iron protein to the P cluster. Instead, this goes through what's called a deficit spending model, where the electron from a P cluster, from the neutral P cluster, is first transferred to the FeMoCo. So now what that results in is a reduced FeMoCo and a +1 oxidized P cluster. So now the +1 oxidized P cluster, shown here in yellow, is now primed to accept the electron from the 4-iron 4-sulfur cluster of this. Okay, so notice that along this pathway, there's a conformational gating that occurred after docking of the iron protein with the MOFI protein. There's a change in the shape of the protein that signaled for transfer of the electron from the neutral P cluster to the FeMoCo. So now, because this is now oxidized, it is more likely to accept an electron. So the electron then gets transferred over to the now oxidized P cluster to return it to the neutral state. And now the iron protein, the 4 iron 4 sulfur cluster of the iron protein is now reduced, which is signified as yellow. Because this is reduced, this induces a conformational change within the protein, which also triggers ATP hydrolysis to form ADP and formation of inorganic phosphate. The phosphate release, so inorganic phosphate release, is actually the rate determining step here. And once this is released, this causes another conformational change, which then allows for dissociation, of the iron protein from the MOFI protein. And what this conformational change basically means is that it lowers the affinity of the iron protein for the MOFI protein, and then it goes away, and so that the ADP can be replaced with ATP, and the iron sulfur cluster can be re-reduced. Right, so recall that all these steps are only transferring one electron to the FeMoCo and ultimately to the dinitrogen. Right? This cycle has to occur 8 times, and each cycle there's hydrolysis of 2 ATPs and transfer 1 electron to the site. And so this happens to happen several times before the dinitrogen is actually reduced to ammonia. All right, so recall, let's look a little bit more at these conformational changes. So the conformational changes are also important for tuning the redox potential so that there can be electron transfer can go smoothly to the dinitrogen. So recall, this is the electron transfer pathway where a ferredoxin is going to reduce the iron protein, okay? And then once this iron protein is reduced, it can then dock with the MOFI protein, and this electron actually transfers through the P cluster to the FeMoCo, and then that ultimately goes to N2. But remember that there is a sort of a dilemma here, right? So while the ferredoxin reduction potential and the iron protein reduction potential makes sense for transfer of the electron to the iron protein, it doesn't make sense from transferring the iron protein ultimately to dinitrogen. Right? The relative reduction potentials aren't such that this is a suitable electron transfer pathway. But what we're missing here is that there are several conformational changes that occur from ATP binding that allow for the reduction potential to change. And so the structural basis for this is, so we're looking at the, this is once again the structure of nitrogenase with both the iron protein and the MOFI proteins attached to each other or interacting with each other. So this is the full complex, and this was done using the transition state analog with the ADP and aluminum tetrafluoride anion. And so in this docking model, what is shown is that there are 2 switches which are really important for this. So this first is called switch 1, okay? And the conformational change that occurs here happens when ATP or when ADP is replaced with ATP. So when ATP is bound, this causes this switch 1 to change its location. And what that allows is for docking to the MOFI protein. It also allows for the 4 iron 4 sulfur cluster, once it's docked, to be closer to the P cluster. And that's very important for the rate of electron transfer between the 2 cofactors. In addition, the switch 2 changes the redox potential of the 4 iron 4 sulfur cluster, particularly when it's bound to the MOFI protein. And so what happens is that instead of being negative 300 millivolts, once the ATP is bound and it's reduced, the reduction potential now drops to negative 430 millivolts. And so that now means that this is more reducing than the dinitrogen, and now electron transfer can proceed as expected all the way to nitrogen through the P cluster and the FeMoCo. So what is happening during all this, right? And so this is not a full picture because as as all these have shown, there's still somewhat of a question. While these depict one electron transfers to dinitrogen 8 times, it's still not quite clear whether physiologically if this happens through 1 electron transfers or 2 electron transfers. So this is not a complete picture of the mechanism of dinitrogen reduction, but this is based on what intermediates have been observed during turnover and what substrates, what alternative substrates can be used to ultimately form ammonia. And so what's known is that there, for instance, dinitrogen, which is in the 0 oxidation state for both of the nitrogens, right, can ultimately be reduced to different 2 electron reduced intermediates, including diamine and hydrazine. And so these have been seen as products during turnover in low concentrations, or they can be used as alternative substrates to ultimately form ammonia. And so that's the evidence that these are intermediates along the dinitrogen reduction pathway to form ammonia. And so we can think of, and while these are in 2 electron segments instead of 1 electron segments, we can think of these at least some of the intermediates, where dinitrogen is reduced to diamine. This is then reduced to hydrazine, and then this is ultimately reduced to break the NN bond and form 2 molecules of ammonia with a -3 oxidation state. And so we can think of that as just subsequently breaking first the triple bond to a double bond, the double bond to a single bond, and then finally breaking the single bond to form the 2 ammonia molecules.